dysfunction resulting from excess inbreeding. The signs of inbreeding depression most frequently cited are reproductive failures. Anything that impacts reproduction is an upfront concern for dog breeders. And I would argue reproduction is a component of fitness, so we're talking about decreases in fitness. Inbreeding-related reproductive failures include lack of libido and low sperm count in males, females, lower rates of pregnancy, smaller litters, fewer offspring, and exhibit poor mothering ability. Poor mothering ability would decrease the survivorship of the offspring. Non-reproductive failures of inbreeding depression can be manifested as a high frequency of immune-mediated diseases, and here's the one I want to highlight. Significantly higher incidence within a line or breed of one or more diseases than is seen in the species as a whole. So as part of inbreeding depression, you get decreases in reproduction and decreases in survivorship, which is a decrease in fitness. That's the negative effect, a decrease in fitness. And the significantly higher incidence of diseases showing up in your line or breed than in the population at large. So let's talk a little bit about what I'm saying. If I'm a dog breeder, and I'm going way back, I've got these dogs, right? Had a nice litter of pups, and I noticed that one of them's like the greatest dog ever. It's like the greatest shepherd you could possibly imagine. The parents, not so good, grandparents, not so good, but this individual is like the best shepherd you could possibly imagine. So I have a trait that's shown up in the population, and it's unique to that individual. So the next thing I would want to do is try to increase the frequency of that trait, in the population over time. Well, if only one of the individuals has the trait, this is probably the individual that I'm going to want to breed. So let's say that that individual that has the trait is a female. Okay? All right, so I have this female individual. I'm going to mate it with a bunch of males in the parental generation to produce the F1 generation, which is the offspring. Then what I'm going to do is take the offspring and begin to breed them with each other to produce, right, the offspring's offspring. Is that okay? This step here, because I'm breeding together the offspring, this is the inbreeding. But because I was interested in the trait that this individual had, I'm engaging in this inbreeding process so that I increase the likelihood of producing individuals with particular traits. So with respect to these dogs, there are going to be some consequences of producing this shepherd line because of the inbreeding that I've engaged in this line. It also means that all of these Australian shepherds that are out there now, they go back to some original individual that started the whole line or lineage. Make sense? This individual is the one I can credit with the trait. Well, that trait was passed on to the next generation through these selective breedings. And today, if I want to maintain the purity of the shepherds, I need to do purebred crosses, which means these individuals I know are descendant from that original individual. So all of the descendants here would be part of a lineage or line that descended from that original individual. what this is saying is within this line, right, or breed, within this line, there are specific genetic disorders that show up in this lineage that you don't see elsewhere. So we need to talk a little bit through that. The cause of this is because I'm starting out with one individual and moving it, the trait through the lineage. I'm picking up particular combinations of alleles that I don't tend to see in the population at large. So this notation is not all that helpful. I did it because I think it's a good way to start, but we need to move to another type of notation that'll make understanding what I've just done a little bit easier. And to do that, we're going to do pedigree. Pedigree is notation to keep track of the lineage. In pedigrees, females are designated as circles, and males are designated as squares. I just remember males are square, and then that helps me remember that. And then this line here indicates mating between these two individuals. Since we're talking about dogs, right, that would be a mating. As you move up to your other classes, in particular genetics, you'll do more with these pedigrees, and typically that line means that the individuals are married. That's because a lot of the early genetics problems were developed in the 20s, so you'll see these problem sets and they'll always say, you know, these two individuals are married. It's not that they have offspring together, it's they're married. But in any event, that line means a mating. This first set of individuals here are part of Roman numeral 1 generation, and then I can number these individuals so that I can refer to them. So 1-1 is the original female that had the trait I was interested in, and then I could also refer to that male individual as 1-2, generation 1, individual 2. All right, I draw a line down from these individuals to indicate the offspring. And let's say that this female had a lot of offspring that were part of generation 2, and this is what she had for offspring. 5 individuals, 2-1, 2-2, 2-3, 2-4, and 2-5. She had 3 daughters and 2 sons, but she had them in this order. A daughter first, followed by 2 sons, and then 2 daughters. Okay? Now, because of what I did in developing this line, I said that we did inbreeding with that second generation. So I'm going to indicate that like this. So I could take these individuals here, and I'm going to mate them together. The double line here means consanguineous mating. Consanguineous, same or together. Sang means blood in the sense of kin, your blood, your kin, your family. Consanguineous is an inbreeding event. Okay? All right? And then this would produce the next generation of individuals. Right, which would be generation 3, 1, 2, 3, 4, 5, 6, 7. Okay? All right, now, let's suppose that in addition to having the trait for shepherding, that this female also happened to be carrying around an allele that's not so good to have. Okay? And this allele here, this little b allele, is deleterious. And by deleterious we mean it reduces fitness in individuals that have 2 of them. They need to be homozygous recessive. Those individuals right, would be suffering a cost to their fitness because the allele is deleterious. Now, this individual, let's say that one is the only one in the entire population on the planet that has that allele. It arose through a unique mutation in this individual, and it's the only one out there. Everybody else is like that. Okay? Now, this would mean that when I mate these individuals together, I'm doing this cross, which, if you remember from MCB, you might have done it called a test cross. The test cross tells me that half of my offspring should be homozygous dominant and half should be heterozygous. So let's go ahead and do that. All right, and let's just take a look at this particular lineage here. This portion of the lineage, their lineage follows, 50% chance, let's say that this individual is homozygous dominant, and that individual is, sorry, when I do this, this will make things a little easier. If I do this, and I do this, because then I've got more offspring. So now I'm doing this cross in the F1. The F2 generation should be 25% homozygous dominant, 25% homozygous recessive, and 50% heterozygous. So let's do that. 25%, 25%. Now, this allele is the only one on the planet, but because individuals are engaging in inbreeding, there's a chance that I can pair up two copies of that allele. And by copies, I mean they were copied from the original ancestral DNA molecule that had this. Mom copied this DNA molecule into the next generation, and by chance, these two individuals got copies of the original little b allele. They then passed on these copies to the next generation, and this individual ended up being produced as a homozygous recessive for an allele that is not common in the population. If these individuals had bred with someone outside the lineage, called outbreeding, the chance of producing this homozygous recessive would have been 0. Couldn't have produced that individual. But because these two individuals had copies of these alleles from mom, and because they're mating together consanguineously, there's an associated probability of producing the homozygous recessive. That is to say, significantly higher incidence within a line or breed of one or more diseases than is seen in the species as a whole. Here, this individual you would never see in the species as a whole because this allele is too rare. Does that make sense? So what inbreeding does is it increases the likelihood of pairing up these deleterious alleles that are identical by descent. These two little b alleles are copies of the same little b allele. They're identical to each other by descent. They were copied from the same molecule. What inbreeding does is increase the likelihood of pairing up these deleterious recessive alleles. That's inbreeding depression. This is the consequence of the inbreeding. It's the increased likelihood of generating these individuals that are homozygous for deleterious recessive alleles that run in the lineage but no place else. Okay? In any domesticated animal, including dogs, I would expect to see a lot of these traits, these deleterious traits, because of the inbreeding that's taken place within the lineage. I'm thinking of like Labrador retrievers with kind of that hip dysplasia that they have, or the little pug dogs that their faces are squished, and they have all kinds of problems, respiratory problems and nasal problems, or the little wrinkly dogs that you have to clean out their folds every day, or otherwise they build up these anaerobic bacteria that cause problems in there because the folds close off the oxygen supply from the outside world, or those little hairless cats, right, that I guess you have to put cream on them every day. I mean, all sorts of issues that these domesticated animals have because of this inbreeding that took place during the lineage and continues to take place during the lineage as you maintain the purity of these Australian shepherds or whatever else you're dealing with. Okay, so I would just want to go through and do show you how one of these calculations would work. Okay, so I'm going to do this first one here as a pedigree. Now, for some reason in this evolutionary biology textbook I stole this figure from, they use shapes for alleles instead of letters. I think that's kind of interesting, but what we're going to do is a Lucky Charms approach here. So there's the green clover and purple diamonds or whatever else. We're going to do red triangles and black diamonds. So this is our individual that we're interested in. The individual here has two alleles. I want to know the likelihood that this individual has two alleles identical by descent. Two alleles identical by descent. Well, one possibility is that mom transmits the red triangle to this individual. The likelihood of that happening is 1/2. The likelihood of transmitting the red triangle to this individual is 1/2. And then the likelihood that this individual inherits this red triangle is 1/2. And this red triangle is 1/2. Now, all of these events have to happen together. This has to inherit the red triangle, and he has to inherit the red triangle, and